Okay, good morning. We're going to continue notes today. The last thing we left off on our outline from yesterday is on the cells. We left off just briefly going over what a chloroplast was. So today we're going to continue our notes, and this time we're going to review microscopes. Specifically, we're going to get into light microscopes because those are the microscopes that Brandon High School has and that we'll be using in this class. So in this class we'll be using a compound light microscope. A compound light microscope is a microscope, obviously, that shines light through the specimen, and it's called compound because it has two lenses, one on the ocular lens and one on the objective, to magnify an image. Magnification means make the image appear larger. Okay, so there are 12 main parts of the microscope. So on your chart, please write in the function of what these parts do, and then after that we will look at what these parts look like on the microscope diagram. So our first part that you need to know is called the ocular lens. This is also called the eyepiece, and that provides a magnification. There's a lens up at the top of the microscope, and that lens is always going to be a 10 times lens. The body tube is next. The body tube is going to be a tube-like structure at the top of the microscope, and its purpose is to separate the two lenses. It separates the eyepiece lens or the ocular lens from the objective lens. If those lenses aren't at a proper distance from each other, the microscope will not focus properly, and so that's what the body tube does. Number 3 microscope part is the nose piece. The nose piece is a rotating device. You turn it. You can turn it both ways, and it will change from low power objective to your medium high or high power objective. After that, you have your objectives. There are going to be 3 objectives on our microscopes. There's going to be a 10x objective, a 40 times objective, and a 100 times objective. There is going to be one other notch on our nose piece, but the objective is not there. All right, next piece is the stage. That is a flat platform to place the slide. Next one is the stage clips. The stage clips obviously hold the slide in place. Then we have our diaphragm. The diaphragm is an important part. It adjusts the amount of light to the specimen. You can make it get brighter or lower by a little knob. It's underneath the stage. Next one is the light source, and that's just a light that is provided for the specimen. Next piece is the base. The base supports the microscope, and it's one of the pieces that you use to hold and carry the microscope with. The next part is the arm. The arm is the handle for when you are carrying the microscope. All right, and then the last 2 microscope parts are the adjustment knobs. The coarse adjustment knob is the bigger knob, and that is the main focus knob that you will use first. So that focus knob you would be using on a low power objective. And then lastly is the fine adjustment knob. It is a smaller knob, and that is for specific focus. You would only use the fine adjustment knob on a higher power. I will leave this up here for another 20 seconds or so you can catch up on your writing down of the functions, and then we will move on to the diagram. Okay, so here is a typical diagram of a microscope. Let's start at the top right and work our way counterclockwise. So the first part is going to be the ocular lens, also known as the eyepiece. And again, remember that this is what you look through to see your specimen, and it does include a 10x lens up there. All right, moving down, number 2 is going to be the arm, and you use that as a handle to carry the microscope with. Number 3 is the stage, and that will hold the slide, and that moves up and down as you focus your specimen. Number 4 is the larger focus knob. It's called the coarse adjustment knob, and you use that coarse adjustment knob only on the lower objectives. Number 5 is the fine adjustment knob. It is a smaller knob, and it is only used on a higher power for specific focusing. Number 6 is the base. It supports the entire microscope, and that is also what you would hold onto when you are carrying the microscope around. Number 7 is the light source. Number 8, very important piece, it is underneath the stage, and it provides a different amount of light to your specimen. So you would turn this knob right here, left and right, and that would make the specimen brighter or make it darker. Sometimes the darker it is, the more clear the image will appear. Number 9 are the stage clips. Our microscope has a mechanical stage clip, so it's going to look a little bit different. Number 10 are the different objectives. So you have, for our microscopes, a 10x objective, a 40x objective, and a 100x objective. They are labeled on there. The 10x objective is shorter, the 40x is a little bit longer, and the 100x, which we will rarely use, is the longest one. All right, number 11 is the nose piece. You can also call it the revolving nose piece. And so this thing twists so that you can change from the low power to the high power. And then lastly is the body tube. And this is just a tube that separates the lens up here from the lenses down at the objectives. And those are the 12 parts to the microscope that you're required to know. Okay, the next section of our notes is going to cover microscope technique. So we're going to go over this basics of how to carry the microscope, how to make a wet mount, in case you've never done that before, how to focus the microscope on high, so that you know what you're doing in this class, and then also how to put the microscope away and clean up. Okay, first part is carrying the microscope. As you can see in the picture, please always support the microscope by holding the base and carrying the microscope by the arm. It is not efficient enough to just carry it by the arm, because then you would probably be swinging the microscope and you can easily damage the objectives. Okay, next part is how to make a wet mount. A wet mount is when you have to make your own slide from a specimen that was given to you or that you brought in. So first you would obtain a glass slide. You would get your specimen that you would like to look at. You would need a cover slip, which is a plastic little piece. So the first thing you would do is put your specimen on the slide, and then you want to add a small drop of water. And so you can see the water droplet on the slide in the picture. Then you want to add your cover slip at a 45-degree angle. And so once you get that angle, you just want to slowly lower it on top of that drop of water, and that should minimize the air bubbles that would show up on your view. All right, the third part we're going to go over is how to focus the microscope to a higher power. First, you're going to put your slide on the stage using the stage clips and get it nice and secure. And usually you would make sure your specimen is in the middle of the where the light will shine up on it. Then you're going to use your nose piece, and you're going to put the microscope actually on low power first. So turn the nose piece, get it onto the lowest power, which is the short objective. Then you're going to use the coarse adjustment knob. That is your main focus knob on low power. You're going to rise that stage up until you see the image clearly. The only reason you wouldn't find it on low is because it's not on where the light is shining up through it. So now you are focused on low power. So to move it to high power, you're going to use the nose piece, turn it to the next power. You don't want to skip from low power to the highest power. You want to go to the next longest objective. Before you go on to high power, you need to center your image on your view, because otherwise you're going to be focusing on to the right of it or to the left of it, and you're going to miss it. So once it's centered, then turn it to high power using the nose piece. And then you want to use the fine adjustment knob. You should be able to see it. It might be blurry. Use a fine adjustment knob, which is going to specifically focus it into more clear view of it. and then you should be all set. Tomorrow, we're going to watch a quick little YouTube video on how to focus a microscope so you can see it visually. All right, some problems that usually come up when we're doing a microscope lab. First one is, it's blurry, I can't see it. So you need to use the coarse adjustment on low, and you have to make sure you're using the fine adjustment on high power. Sometimes that's the problem, is you're just not using the right objective lens, you're missing the focus, something like that. Otherwise, maybe the lens is dirty. So we have special wipes, and only these special wipes are the only thing you would wipe down your objective lenses or the eyepiece with, or even the slide. There's special lenses that will not scratch it. So use those, wipe them off, and then sometimes actually that makes it clearer. Another problem is the slide, or the objective is hitting the slide. I think that's written wrong. There really isn't anything you can do. Maybe your specimen is too big and it's too thick or something like that. So for most likely you will have to change microscopes or thin out your specimen. And then a really big problem is it's too dark or it's too light. You can't see it very clearly, and that's when you have to use that diaphragm, because a diaphragm will make it darker or make it lighter, and it makes a huge difference. Okay, so at the end of the hour, when we are cleaning up for lab, these are the steps that you would do to your microscope. First, you need to make sure that you remove your slide off of the stage, and you return it and clean it off if necessary, and put it back into its proper spot on the back lab table. Then you need to turn your nose piece and put the microscope on low power. You do not want to leave the nose piece on high power. Another thing, your stage needs to go all the way down. So you're going to use the coarse adjustment knob and make that stage go all the way down as far as it can go. You're going to take the cord and wrap it around the microscope, as you can see on the picture on the left. That is correct, one. And then you also want to align your body tube to the center, because our body tube can twist around. And so you want to make sure that's aligned so it won't hit anything as you're carrying it back to the cupboard. And so you are allowed to go in the back room to put your microscope back. And you can see on the right that the cord isn't properly wrapped around, it's on high power, and the stage is up. So those are the reasons why that microscope on the right is wrong. Okay, the last part of the notes is making sure that you know how to draw and label on our drawing sheets. So to draw an image under the microscope, you're going to want to draw your entire field of view. So as you look through the microscope, what you see in that circle under the microscope is what you draw on your drawing sheet. So you're drawing the entire field of view in the circles. Also, at the top of your picture, there should be a line provided for you. You're going to title what image you are looking at. And then also you're going to jot down the magnification. So remember, the magnification is the ocular lens times whatever objective you are using. So if you are using the 10x objective, then it's 10 times 10, and that would be 100x, as shown in this example of this amoeba. Otherwise, if you're using medium power, it would be 40x times the 10x ocular lens, so that would be a 400x, and then sometimes you might end up getting to a 1000x. When you are labeling, you can see on this picture it is labeled good. The lines are nice and straight coming off of what you are pointing to, and you are always labeling it outside of the circle. You would never write nucleus, for example, inside that circle. It would have to be on the outside. You don't want to, there should be no labeling on anywhere inside. All the labels are on the outside of the circle. And that finishes up the notes.